Hello, welcome to Callista's bio class. Now, this video is for most of us that are preparing for our post UTME examination and that have put up most Nigerian universities like Uniben, Unizik, UNN, UI, and so on. So if you know that you have been reading, studying, I'm also here to guide you, to help you out, especially in the subject biology. So let's dive right into some biology revision questions that would help you to ace your score in this post-UTME examination. Alright, so I hope you can see the question on your screen. Let's go to question number 1. It says, in which of the following organisms does each cell combine the functions of nutrition, reproduction, and growth? Option A, we have rhizopus. Option B, we have Dryopterids. Option C, we have Brichamenium. And option D, we have Spirogyra. Now, look at the question. You have to understand the question very well before you can attempt or even know the options to go for and those to neglect. Remember, I said something in my other video that if you want to know the answer to questions and work with time, because most examinations deal with time, there is a timing. Just know the questions that you should remove the irrelevant options and go for the ones that you should calculate between. Now, this question is indirectly telling you, look for that plant or look for that option that is a unicellular, because it is only in unicellular organisms, unicellular, that is what we refer to as one cell organisms, that you can actually have a cell, they combine all functions. But in multicellular organisms, there are many cells and they have similar cells coming together to perform a function. But in unicellular, just one cell doing all the functions. So indirectly, they're asking you which of these is the unicellular organisms. Do you understand? That's also what this question is all about. So if you look at option A is what? Rhizopus. What is a rhizopus? It is a fungi. Remember, most fungi are multicellular. Aside the yeast is a unicellular fungi, just one cell. So if this was yeast, we would have gone for this option. But because it is a rhizopus is a multicellular fungi. Hence, it has many cells. You cannot have a case of one cell combining all these functions of nutrition, reproduction, and growth. So therefore, you're not going to go with this option. Now, option B says Dryopterids. Dryopterids are ferns. They are under the kingdom Plantae that belong to the division called pteridophytes, where we have the fern plants. And these are already multicellular, they are not unicellular. Remember, they're actually asking you for a unicellular plant, a one cell plant that can carry out all functions. Do you understand? Now, option C, Brichamenium. This is a bryophyte. Bryophyte like moss plant, liverworts. These plants are known to have structures that looks like roots, stems, and leaves, but they are not true root, stems, and leaves, and they are also multicellular, so we can't go for this. Now, option D, it's what? A Spirogyra. If you've been studying and you know your biology, you will know that Spirogyra are actually unicellular, but they exist as filaments. Meaning, when you look at some unicellular cells, they can exist in colonies, in filaments, or free living, or they can exist in cells can exist in different forms, not just unicellular. Cell can exist in different forms. A cell can exist alone, unicellular. Do you understand? The free living, like when you look at some amoeba cells. You can also look at some cells existing in colonies, in groups. Do you understand? Like when you look at Volvox, Pandorina, Eudorina, these are cells that occur together in clusters. But it doesn't mean that they are multicellular, because one cell can still stay independent of the other cells. But because they are in colonies, they have more effects. They can exert more effects. They can produce food. They can fight their enemies better. It's just like one man standing alone against a multitude. Do you understand? But they are all unicellular cells. Then Spirogyra is a unicellular cell, but it is known to exist in filaments. So if you have just one cell out of the long chain or long filament, one cell can on its own independently function as as a unicellular organism it is, and still carry out the functions of reproduction, growth, and what? Nutrition. So this tells that option D is actually the correct answer, which is what? Spirogyra. All right. Now let's move on to the next question. All right, you can see the next question on your screen. So let's go. Question number 2. It says, bacteria multiply rapidly by means of A, budding, B, fragmentation, C, binary fission, and D, spore formation. Now, bacteria are known to multiply in, a bacterial cell, depending though, some can use 30 minutes to what? Increase. And they always do this by what? Binary fission, whereby one bacterial cell divides into two, and then 2 into 4. So binary means actually 2. Fission has to do with division. So they divide into multiples of 2. Do you understand? Some bacterial cells can do this under 30 minutes. Some bacterial cells can do this under what? 20 minutes. Some more than 30 minutes. So that's why a bacterial cell easily rapidly increases in size. But option A says budding. Now budding is not the answer, but what actually is budding? Now budding is a type of outgrowth whereby a young plant or a young organism that is still growing out from the parent organism, stays there, just like the way you see a little branch coming out from a tree trunk. So it comes out, it continues towards grow. When it can stand on its own, exist on its own, it breaks off from the parent and starts existing. Example of organisms that carry out budding are hydra. Then we also have fragmentation. Fragmentation just simply means to divide into what? Parts. Most organisms that can undergo this kind of fragmentations are something like amoeba. They can split. Then we also have what? Binary fission. That is actually the answer. Then spore formation has to do with most plants that like the rhizopus, the fungi, them some plants can also actually carry out spore formations, like moss plant, bryophyte, ferns, whereby they have their sporophyte stage that bears the spores inside a sac. So when these spores stay there, they ripen and mature, and they burst open and are carried by the wind. If they fall on a fertile place, they begin to what? Germinate. The same thing happens to most fungi. But a bacterial cell, it doesn't undergo spore formation, but it increases rapidly by what? Binary fission. So option C is the answer. All right, let's go on to the next question. Question number 3. All right, welcome back. So let us look at question number 3, and it says, you can see that on your screen. It says, after a meal of yam has been digested, the highest concentration of glucose is to be found in the... Now, where do you find the highest concentration of glucose after a meal of yam? You all know that yam is a carbohydrate, right? So where do you have the highest concentration of these carbohydrates broken down to glucose? Where do you have it? Option A says hepatic artery. Option B, pulmonary vein. C, hepatic portal vein. D, posterior venal cava. Now let's look at option A, hepatic artery. Now the hepatic artery is just an artery that is connected to the liver, and it's there's something about the liver you need to know. The liver is one organ that gets blood directly from two sources. It gets blood directly from the direct arteries are those blood vessels that do what? That bring blood to every other organ from the heart. It brings blood rather from the heart to every other organ. You understand? From the heart, it distributes blood, but the veins return what? Blood back to the heart. So anytime you hear hepatic, it has to do with the liver. Now, what does the hepatic artery does? The hepatic artery, what it does is that it brings blood from the heart to the liver, because hepatic has to do with liver and artery has to do with bringing blood from the heart to every other organ. So hepatic artery brings blood to the liver. But just remember what I said, that the liver is one organ that gets blood to it through two ways. Now, through the first one, which is the hepatic artery, and there's another one we call the hepatic portal vein. Now, the hepatic portal vein, from that word portal, what does portal mean? Portal is like a gateway, a passage. So when the blood comes in through the hepatic portal vein, The hepatic portal vein is one vein. Normally, vein should return blood from any organ back to the heart. But this vein is not a true vein. You understand? Most people, it's actually discussed or debated that it's not actually a true vein because it doesn't behave like other veins. What the other veins do, other veins actually take blood from organs to the heart. But this particular vein, hepatic portal vein, takes blood from the mesenteric organs like the intestine, after it has digested food, and the food has been the food now wants to be absorbed. Now the liver, the food will pass in through the liver. So what happens is that some of this digested food will get to the liver, where the bile and other things would be secreted, and some other toxic were taken away from the food. So in essence, the hepatic portal vein brings blood to the liver. Do you understand? And the hepatic and another vessel that brings blood to the liver is what we refer to as the hepatic artery. So the question here now says, after a meal, the one of course that should have the highest concentration is the hepatic portal vein. Why? Because this is one that is gotten from the intestine, where the food has just been absorbed, and they are getting the food that has just been broken down into the lowest form, which is glucose for carbohydrates, amino acid for protein, and then fatty acid and glycerol for fats and oil. So of course, this hepatic portal vein has the highest concentration of glucose, because it's just coming in right from the villi, the part of the small intestine where absorption of food occurs. Do you understand? So that is the answer to this question, option C, which is hepatic portal vein. While the posterior vena cava are the part of the lower chamber of the heart, where every organ below the heart returns to the heart through the posterior vena cava. Then we also have something called the superior vena cava, where every organ above the heart returns to the heart. Every organ above the heart, the blood from the vein returns to the the major vein, because the vena cava is a major vein. So the blood from every organ above the heart returns through the superior vena cava, while the blood from every organ below the heart returns back to the heart through a major vein we call posterior vena cava. Do you understand? All right, so the option to this question number 3 is hepatic portal vein. All right, let's move on to the next question. Question number 4. All right, so question number 4, it is on your screen. I'm sure you can see that. Now let's read. It says, the structure that is most commonly identified in all living cells under the light microscope is the A, mitochondria, B, chloroplasts, C, nucleus, D, ribosomes. Now, what is a light microscope? You need to know that the light microscope is that microscope that is, we refer to it as, actually we have the very high powerful microscope referred to as the electron microscope. Do you understand? Then this light microscope is another one that is close to the electron, but it is not very powerful like the electron microscope. Actually, the light microscope is divided into two. The simple microscope, the one that uses only one lens, it doesn't have an objective lens. And then the second type of light microscope is the compound. From the name compound, it combines two lenses. The very first lens, which we refer to as the eyepiece, through which the observer using the microscope uses, and then the other the revolving lens, the objective lens that we can revolve to either use the high power objective or low power objective lens. So the compound microscope uses two. Do you understand? Why the simple light microscope uses one objective lens. So that tells you that the compound microscope has a higher power, followed after the electron, which has the higher electron microscope, which has the highest, because it uses tiny electron particles, unlike the light microscope that uses light source, either a natural light source or an electric power source. Do you understand? So now this tells you that in order to see any very tiny objects like viruses, you must use an electron microscope. But if you're using just a light microscope and you want to view the cell, Now, there's some cell organelle that you will easily see without any, special technique of staining the cell. You're just going to see it, like the nucleus is one of them. But something like the ribosomes, you need to use a special staining technique before you can see the ribosomes, identify it like they are ribosomes. Then, if you look at which other option do we have, we also have option A, mitochondria. You can see the mitochondria, but you won't see it. It will be very distant. Then, of course, the chloroplast. Remember that you can only see the chloroplast in plant cells. It is that part that contains the chlorophyll. But the question here says the structure that is most commonly identified in all living cells. They did not specify either plant cells or animal cells. So this tells you that the answer to this question is what? The nucleus. You can easily see the nucleus. You remember, yes, some of you might be thinking that the nucleus is not that big in animal cell, because the what's it called? The nucleus is not that big in plant cell, because the plant cell tend to have what? A large vacuole. But remember that no matter the nucleus is not that big, but it's still big enough that it can be easily seen. Though the vacuole of the plant cell is big, that it shifts the nucleus to the side, but still the nucleus can be viewed. Do you understand? But when you're looking at some structure like the ribosomes, you won't easily see it. The mitochondria, you will see it a little. Do you understand? But the nucleus, you must easily identify it. Do you understand? When using a light microscope. So the answer to this question is option what? Option C, the nucleus. All right. So that being said, let us move on to the last question, question number 5. All right. So let us look at the last question, question number 5. It's on your screen. All right. Let me read this. It says the possession of scales. That's the first one. The possession of scales, laying of eggs with shells, and bony structure of the head are characteristics shared by, I repeat, the possession of scales, the first characteristics, the laying of eggs with shells, that's the second one, and bony structure of the head are characteristics shared by. Option A says birds and reptiles. Option B says fishes and birds. C says reptiles and fishes. And then D, birds and mollusks. Now, you don't need anybody to tell you that the chordates, that is animals with backbones, that are called, they belong to the phylum Chordata and subphylum Vertebrata. They have 4 subphylums under them. We have Pisces, that's where we have fishes. We have amphibians, where we have the toad and frog. Then we have reptilia, where we have reptiles. And then the last one, mammalia, where we have mammals. Now, one of the characteristics of some organisms that are known to have scales, scales on their body, you see them in reptiles like snakes, lizards. Then you also see there's that scales, you don't see it on mammals. There are mammals that covered with hairs or furs. Then you also see you don't see scales on amphibians. Amphibians have a moist body that could look a bit towards rough or smooth. Do you understand? Like in the frogs, it's smooth, while in the toads, it's a bit, bumpy. Then when you see scales, another place you can see scales are in what? Birds. If you look at the birds, they have what they call feathers on their body, but on their legs they have scales. So another option, another characteristics there, we have they possess laying of eggs with shells. Now, mammals don't lay eggs, or sorry, rather, we have mammals that lay eggs. Birds, we also have the ones that they don't lay, they just give birth to their young ones alive. Because we actually have 3 types of mammals: the egg-laying mammals, we call them the what? Monotremes, the marsupial mammals, and the placenta mammals. Placenta mammals are those ones that they feed from their, the placenta of the mother, bearing the organism or the young ones. Then the marsupial mammals are those ones that they give birth to their young ones alive, but premature. Then they put them in that sac-like structure called the more like the marsupial. So that sac-like structure called the pouch rather. So examples of when they put them there, they put them there to develop, like in the kangaroos you see them. So these are marsupial mammals. So we actually have egg-laying mammals, but they don't lay eggs like the way the shell would. So, but in these birds and lizards, they have the ability to lay eggs that have a hard shell. Then frogs or amphibians, which are frogs and toad, actually lay eggs, but they don't have shell wrapped around or covering them. Why? Because most organisms that lay eggs in water don't need shell. The water there is enough to make the sperm swim to the egg and to not make the egg dry up. But most organisms that lay their eggs on land need to lay an egg that is covered with a shell to protect the egg, and then also so that the egg does not dry up. Do you understand? From protect the eggs from desiccation or drying up. Do you understand? And most of the times they lay fertilized what eggs. Do you understand? So the eggs has to just be in a warm place where it will be incubated and then it's ready to be hatched. Do you understand? When it's ready. So this tells you that, and then if you look at the other characteristics, says the bony structure of the head. The head of reptiles and most birds have a bony structure. Do you understand? So this tells you that the answer to this question is what? Option A, birds and reptiles. So that being said, we come to the end of those section, biology revisions on some possible post UTME questions. I'll still try as much as possible to still share more videos with you so that you can get you can get properly prepared before your post UTME examination for this year 2024. And I wish you all the best, and I pray you get that score that will give you your dream course into the Nigerian university of your choice. Stay tuned to Callista's Bio class and see